Good evening. Welcome to the Puya lecture series. My name is Minu Varzaga, and I am the president of Puya, Professional Organization of Iranian-Americans in Atlanta, which is a branch of Persian Cultural Center or Kanun Farhangi Iranian Atlanta, a non-religious, non-political, independent organization. Initially, I would like to extend my gratitude to our world-renowned learned scholar and professor and head of the Department of Smell and Taste, Dr. Richard Doty. It is an honor to have you speak for us tonight. I also would like to thank the participants in this lecture series and our supporters and the board of directors of Puya, who are all highly educated and professional in different fields. Puya's board is different from that of Kanun. However, it is related to Kanun and is a branch of Kanun. Puya seeks to celebrate the heritage and achievements of all Iranian Americans, as well as our American and international professionals. Puya works hard to uphold the two pillars of its mission. One, to provide an opportunity for Iranian Americans of generation 1.5 and 2 professionals, and help them to network and prosper. Number two, to promote a positive image and counter the negative media depiction of Iran and Iranian professionals. The reason that the medium of communication of this organization is English is to attract our Iranian American professionals, as well as our American and non-American and non-Iranian people to our lecture series, which are scheduled monthly. A very, very brief introduction of Dr. Doty. I said it to our friends before the meeting that I wanted to actually print out Dr. Doty's CV. Then I went to the room. My printer was not doing anything, and because I was out of paper, I was out of ink, and there was a pile of paper, on the floor, and 120 pages were there, and it was not, done yet. So that is who Dr. Doty is. Dr. Doty, Dr. Richard L. Doty, PhD, is a professor of psychology and ENT with a specialty in head and neck surgery at the University of Pennsylvania. He has also been the director of the University of Pennsylvania's Smell and Taste Center since 1980. Dr. Doty is considered a world-renowned researcher in the field of olfactory functioning and dysfunction. Dr. Doty is a pioneer in the development and validation of practical quantitative tests of olfaction, including the University of Pennsylvania Smell Identification Test, or UPCIT. Education and training. Dr. Doty received his Bachelor of Science degree from Colorado State University in 1966. He was awarded a Master of Arts in Experimental Psychology with an emphasis in psychophysics from California State University, San Jose, in conjunction with NASA's Ames Research Center in 1968. Dr. Doty earned his PhD in comparative psychology and zoology from Michigan State in 1971. He was a postdoctoral research fellow at the University of California, Berkeley from 1971 to 1973 in the field of behavior and chronology, and was advised by Dr. Frank Beach. At the Monell Chemical Senses Center, in Philadelphia, Pennsylvania. He was a postdoctoral fellow from 1973 to 1974, and then director of the Human Olfaction Section from 1974 to 1978. Dr. Doty has published over 400 papers in peer-reviewed journals related to olfactory and gustatory function, and is the editor or author of 9 books. He is the editor of the 3rd edition of the Handbook of Olfaction and Gustation. Wiley Blackwell, 2015. Considered by experts the chemical senses field to be a crucial text. Among his other books are The Neurology of Olfaction with Christopher Hawkes, Cambridge University Press, 2009, and The Great Phenomenon Myth, Johns Hopkins University Press, 2010. Dr. Doty has received multiple awards. In 1996, he received the James A. Shannon Award from the National Institute of Health. Dr. Doty received the Olfactory Research Fund's Scientific Sense of Smell Award in 2000. In 2003, he received the William Osler Patient-Oriented Research Award from the University of Pennsylvania. In 2004, Doty received the Society of Cosmetic Chemists Service Award. A year later, in 2005, he was recognized by the Association for Chemoreception Sciences with the Max Mosel Award for Outstanding Achievement in the Chemical Senses. Okay, Dr. Doty, I think that would be enough, for our guests to actually get to know a little bit about you. We are honored today, and I must also, give my gratitude and appreciation to Dr. Pourezai and Dr. Alavi, who were actually the sources of this, this lecture tonight, In a Zoom meeting, I got to know Dr. Doty through Dr. Pourezai, Dr. Moin, and Dr. Alavi, and I am grateful to all of them for actually making this night possible. Dr. Doty, it's from let me thank you. From PUYA, professional organization on behalf of PUYA, professional organization of Iranians in Atlanta, and also myself, Dr. Doty. We are all ears. Thank you so much for actually giving us this honor tonight.
Thank you very much. It's really a pleasure to be here and an honor to be here. It's really wonderful, and I've learned so much. Shima Moeen has been very helpful, as have all the other colleagues that you've mentioned, and she helped me redesign my presentation so it's a little bit more modern and clear. So I'm grateful to her as well. I'm going to be talking about the science of smell and taste and its clinical applications. And a few disclosures. Mainly, I'm actually the president of a company called Sensonics International, which manufactures smell and taste tests. Years ago, no one was interested in these tests. I actually got the university to sign over to me, which is rare today. So I was able to start a company that's been able to maintain some degree of testing throughout the world. So today I'm going to talk a little bit about the anatomy and physiology of these two sensory systems, primarily with the focus on the sense of smell. I'll provide a basic overview of how smell and taste testing is performed in the clinic, which has really been my forte, largely in the latter part of my career anyway. And then finally, I'll describe examples of disorders with losses or distortions of the chemical senses. A little bit about the Smell and Taste Center. We were funded back in 1980. It's only one of four such centers set up by the National Institutes of Health that still survives. Our mission was threefold: to provide clinical evaluation, treatment, and counseling for patients with smell and taste disorders, provide facilities for an intellectual focus for research in basic and applied science related to smell and taste, and then also provide training and educational support for a variety of people as well. We've published a number of papers over the years, and I've been fortunate to be involved with some really great colleagues in regards to this. So in recent years, I don't have 2021 in here because I would have to go up 4 slides. The research that's been going on since COVID is amazing. There's probably about 12 or 13 other papers now if you just look under COVID and smell, and it really is quite remarkable. But as you can see, that interest in the senses of smell and taste, at least in the fields of ear, nose, and throat and neurology, has been dramatically increasing over the years, as have the books that most of which I've been involved with have come out. I think the average person really doesn't think about how important the sense of smell is to them. But in reality, it's very important. And I always give this slide to just reiterate why this sense is important. Do you see my pointer, by the way, when I use the pointer or not? I guess not. Okay. So certainly the flavor of foods and beverages, nutrition, quality of life, safety, a disproportionate number of the elderly die in accidental gas explosions, fires, and poisonings. And as we get older, as you'll see later, we tend to lose our sense of smell. There are many other aspects of communication, occupation. You can imagine if you're a firefighter or a plumber and you can't detect leaking natural gas. We know that the U.S. Armed Forces, if you can't smell, you're not officially allowed to be in the armed forces, although they have a hard time getting people and they sort of ignore that. But it is in the medical regulations that if a person cannot smell, they're not allowed to enter into the armed forces. One of the more interesting things clinically is the fact that smell loss appears to be among the very earliest signs of neurological disorders such as Alzheimer's disease and Parkinson's disease. Indeed, the smell loss will occur up to a decade or more before the motor symptoms in Parkinson's disease. So a little bit about the anatomy and physiology. We start out in life with 6 to 10 million of these bipolar receptor cells that are up here in that circle. Among those receptor cells, there's about 400 different types or classes. And a particular odor that we perceive, let's say chocolate, is dependent upon some subset of those receptors firing synchronously or together in an appropriate manner. We'll see clinically when some of those cells are damaged, then the chocolate smell, for example, will be distorted. The receptors are located way up at the top of the nose, as you see in the top there. And the area, about 6 or 7 square inches. I'm not sure how many centimeters that is, but it's a large area that contains the receptor cells as well as a number of other cell types. These cells are unique in a variety of ways. One is that they can replenish themselves when they're damaged, to some degree anyway. Also, they're a major conduit of viruses and xenobiotics and nanoparticles and things like this up into the brain as well. So there's been many enzymes in the mucus to protect us from that and so on. But it is a way that things can get into the brain and bypass the blood-brain barrier. So those cells extend up through what we call the cribriform plate, up into the brain cavity, into the olfactory bulb, which you see at the bottom there. And then it's a complex series of cells that integrate information, modulate information, and so on. I don't get into the details here, but from there, the information is passed on to areas of the cortex, mainly the piriform cortex, the entorhinal cortex, and so on. And I'll have a slide to show the complexities of the system at that level. The mucus in that region comes from what I call Bowman's glands largely, and it's quite unique. There's a lot of enzymes in there that protect against cell molecules getting up into the brain, detoxifies molecules. And that's different than the surrounding respiratory epithelium, which is the lower part of the nose. So these are just the main structures. They're in some mammals, they have what's called cranial nerve 0 or the nervus terminalis. It's a different nerve. We have that, but we don't know its function. And then the feeling in the nose, if you experience menthol, the coolness, the sharpness of menthol, that's through a different nerve called cranial nerve 5 or the trigeminal nerve. And olfaction is considered to be the olfactory cranial nerve 1. And some animals have other structures in the nose that are sensitive to odorants of various types or chemicals of various types. We as humans lack those structures. So it's quite interesting. On the left here, you'll see with an electron microscope what the epithelium looks like. In the bottom half of A here, you'll see what the cilia are that extend out from the receptor cells. And at the top half, you'll see the very short cilia of respiratory epithelium. The cilia of the olfactory receptors, remember I mentioned a few square inches, 6 square inches, but if you actually multiply that out in terms of the length and the number of cilia, it's quite large. But we're nothing like the German Shepherd dog. In the German Shepherd dog, if you calculate the number of cilia and the size of the cilia, the length of the cilia, the width of the cilia, the surface area of the cilia is about 1.5 times the surface area of their entire body. So it's not surprising that dogs have a much better sense of smell than we do, although we have a very good sense of smell as well. And in the center, you'll see in B here, just a cross section of what the epithelium looks like. The far right, you'll see those little end there. Those are nerve fibers or the axons that are getting ready to course up from the nasal cavity up into the brain. And there are various cell types that regenerate that you see here, the microvillar cells or the M. There are cells that are dividing and so on. And then the olfactory bulb is the structure, the basic structure of the olfactory bulb is shown on the right. It's basically a laminated structure and very complicated. A lot of information, a lot of sorting of information or integration of information occurs in the bulb rather than in higher areas of the brain. So it just gives you an idea of the complexity, even at the periphery of this sensory system. When you get into central structures, it gets quite complicated. And there are reciprocal relationships between all of these different brain structures that help to determine what a smell smells like. And I won't go through all these different structures, but you can see that the entire brain, to a large degree, is involved in the perception of odor. Taste is a bit different. What we see with taste, we have taste buds, which are shown in the upper right. They're shaped like a rosebud, so the early anatomists called them taste buds. And at the top of the apical area are the receptors, microvilli, that are where the taste receptor, biochemical receptor, is located. But you can see that it's a fairly complicated structure. And those taste buds are found only on this papilla, for the most part, on the tongue. But you see in the back of the tongue, there's a large circumvallate papilla, sort of form a chevron in the back. They have the most taste buds. And then the fungiform papilla in front of that, the edges on the side there are the filiform, are the foliate papilla that folds. And then in the center, the bumps on the tongue are the filiform papilla. If you have a cat and it rubs your tongue, rubs your hand or whatever, licks your hand, it's that sharpness of those filiform papilla, they lack taste buds. In the bottom left here is a picture of a one of the papilla sticking out among the filiform papilla, and the little dots on that papilla that you can see are actually entrances into those taste buds. The given papilla will have a number of taste buds typically. On the right, you'll see the anatomical structure of the projections into the brain from the tongue. So the anterior part of the tongue is innervated by a nerve called the chordotympany nerve that projects into the brainstem. The back part is the glossopharyngeal nerve, and the way back is the vagal nerve. And so the projection of these nerves is orderly and shows a sequence in the brainstem, as you see there, and then that projects up to the thalamus and to areas of the cortex. So I was fortunate years ago, we received funding from the NIH for a smell and taste center. And one of the first things we decided was needed was a simple way of measuring smell. And we didn't know much about it. In those days, everybody was measuring thresholds. They would have to have complex equipment, and they were very concerned about, the number of parts per million or billion. And it was a very onerous task to measure smell function. I happened to have a little laboratory, and I was playing around with stuff and just had people smell a bunch of odors as well as doing threshold tests. And we thought originally we would develop a test to bring people in to be further tested with the more sophisticated threshold testing. And what we found was that the test that we had designed to bring people in was a better test than the threshold test. So first of all, why do we perform quantitative testing? Well, patients aren't very good in recognizing how well they can smell or taste. And without accurate assessment, you really can't tell whether they have a true problem or not. But these are all the reasons to do quantitative testing, just like we do with balance or hearing or vision. You really want to be able to find ways of assessing what is there a problem, the degree of the problem, the nature of the problem. So odor identification was developed, and we were the first really to get involved with this. And we devised this test called the University of Pennsylvania Smell Identification Test, which was a series of booklets that had scratch and sniff labels, so-called microencapsulated labels. People would scrape open the odor and then give an answer, and then indicate the answer on a key at the back. And then when they went through, originally it was 50 odors. We narrowed it down to 40 odors. And then the total score was a measure of their function. And the advantage of this test is that it was simple, it was sanitary, it's thrown out. It could be administered, self-administered in about 15 minutes. Turned out to be very reliable. And then we developed norms based on thousands of people. And so we could, for example, in this case, this is for women. Women tend to have a little bit better sense of smell than men do. But we could, based on the simple score, in an absolute sense, determine if people were normal or normosmia, or they have mild loss, moderate loss, severe loss, or totally anosmic, or if they're avoiding the correct answer. In clinical cases, by chance alone, you have to make a response. There are 4 choices. So if you have no sense of smell, assuming a random distribution, you should score on average 1 out of 4, or around 10 out of 40. And you can see that if you're down to very low numbers, if you miss everything, it means you're avoiding the correct answer. So a very low probability of getting a 0 on a test like this, so-called forced choice test. So we can accurately detect people who are cheating the test. And sometimes we see in head injury cases and so on, they may have gotten their function back, but they continue the legal case going and so on. But when we test them, we show that, in fact, they're avoiding the correct answer. We also can, with this kind of test, look at the relative performance within a given age group. So you can see if you're 80 years of age and you score on average at the 50th percentile, you actually have a moderate loss. But you can see the drop off, and those lines, the middle line is the median, and then the top line is the 75th percentile, and the bottom line is the 25th percentile. And you can see how after the age of 60 or so, the sense of smell drops off dramatically. But again, if you're an 85-year-old, on average, you have severe loss, but it's normal for your group. If you go down that column, you'll see that 49, a score of 17, is the 49th percentile, which is average essentially for an older person. So in this way, we can counsel patients not only in terms of whether they, in an absolute sense, have normal function, but we can counsel an older person that while they have a loss, it's not atypical for them. And this shows this before we did this study, interestingly enough, there was some controversy about the effects of age on smell. And in fact, some people said they had no effect. But as you can see, there's a massive effect, as was shown on the previous slide, where women outperform men, but both men and women start to show a major decline after the age of 60 or so. This just shows various disorders and so on, but notice cheaters. These are Penn students who are said to pretend you can make a lot of money by convincing the examiner that you have no sense of smell. You can see that most of them got a 0 or very low. So they hadn't taken a statistics class, I guess, because they avoided the correct answer. And we were able to catch that kind of behavior, even with bright young people. We've developed other tests using that type of scratch and sniff methodology. This is a test we use for children. Basically, they scrape open an odor, give an answer, and then it rotates around. And when it's totally rotated around, the correct answers show up in little circles. We can add those up. So this keeps the attention of children, and it allows us to evaluate them when otherwise they're not easily evaluated. I had some parents come in, they'd taken their child to all over the world and claimed the child couldn't smell. And so I played a game with this test and gave the child, I think it was probably a quarter for each correct answer, and encouraged him, and he turned out had a normal sense of smell. Parents were very upset, thought I was crazy, and wanted their money back. But it was, we were able to show that child could in fact smell normally. Threshold is a very common way, measuring the lowest concentration, just like hearing, the lowest concentration of a smell. We've developed various tests, but one of the tests are these wands, where when you push forward with your thumb, a felt tip like material comes out, and we have a different concentration. So we go through an algorithm to measure the threshold, and we can have a very reliable threshold. It tends to correlate very well with the upset, or that identification test that I mentioned earlier, but it's another way that is commonly used to measure smell function. And we've also have computerized systems that will automatically calculate a person's test score when they make responses on the touch screen and so on. So you don't have to increase it if they miss it, or decrease it when they get it right in terms of concentration. The computer does all of that. Taste testing, up until recently, there wasn't a very practical means of testing taste, but fortunately we developed on the left here a series of taste strips we call the waterless empirical taste test. There's 53 of these strips, and some are blank, and they have different concentrations of sweet, sour, bitter, and salty tasting substances as well, something called umami, which is monosodium glutamate in this case. And they run that around their mouth, and then we get a total score as well as score for the different materials, different qualities. Then on the right, you see what's called an electrogustometer, since science has recently just started to produce these. But those of you that have ever tested a battery in your mouth and put on your tongue, a lot of people don't reach your age that do this much, but they basically, just as you can determine whether a 9-volt battery is active, it's the same principle. except here we titrate down to microamps and can establish threshold measures using this device. And this goes to show this waterless empirical taste test, just like the UPS, it's self-administered. People will go through a series that's given to them, and then they indicate whether the sweet, sour, bitter, salty, and so on. And then we get total scores, and then there are norms based upon on responses in a normal group of people. Interesting, the front of the tongue is generally more sensitive than the back of the tongue to all taste stimuli, whether they're sweet, bitter, salty, in this case, electrical. And then there you see the age effect that occurs also in taste as we see in smell, young, middle, and older people. And then, of course, different parts of the tongue have different sensitivity. There's sort of an old wives' tale that only the front of the tongue is responsive to sweet, and only the back is responsive to bitter. But as you can see from this, all the tongue areas, to differing degrees, can detect sweet, sour, bitter, and salty sensations. So what are the causes of smell dysfunction that we see in the clinic? And there are many. This is a listing of just some, but the three major causes that we see in our clinic and other clinics that specialize in smell and taste are head trauma, nasal sinus disease, rhinosinusitis, polyps, things of this sort, or viruses. The common cold, believe it or not, is the most common way of losing your sense of smell permanently. Typically, someone's in their 40s. The average age, I think, in our sample of hundreds of people is around 43. They get a bad cold, they get over the cold, they discover that food doesn't taste right, and they discover they can't smell well. Influenza does this, and of course, today, the SARS virus, the COVID-19 virus, really is brought to the fore how a virus can damage the sense of smell. Most cases of return to normal in some period of time. That's true. You know, when we have a common cold, we usually attribute the loss to the blockage in the nose, not to any receptor inflammation or damage, but there may be similar principles occurring between of all viruses and why we can't smell. We don't know a lot about that. But the major reasons for smell loss are the head trauma, rhinosinusitis, and things of this sort, and viruses. A head trauma, if you just slip on the ice and hit the back of your head, there's a movement of your brain, a coup contra coup movement of the brain that can shear off those nerve fibers as they course between the nasal cavity and the brain cavity. And therefore, if something's rather innocuous, otherwise, don't have to lose consciousness or have any evidence with a CAT scan or an MRI of any brain damage, but you can still have loss of smell. Now, you'll see in green, these are various neurodegenerative diseases or neurological disorders that are associated with smell loss. In blue, you see other neurological or psychiatric disorders that are not associated with smell loss. So neurologists can differentiate, for example, between someone with Parkinson's and someone with progressive nuclear palsy based upon smell testing, and that's used in some clinics. I mentioned earlier that over a 5-year period, if you can't smell, you're 3 times more likely to die than if you can smell. And this is after controlling for many variables. We don't quite understand it entirely, but we believe it could relate to the number of different types of viruses that you have been exposed to that have gotten into the brain. It could be related to cardiovascular issues, the blood vessels, things of this sort. It's really a mystery, but this phenomenon has been shown, I think, in 6 studies now. So it's really clear that when we're older and if we lose our sense of smell, we are more likely to have a shorter mortality. Obviously, a lot of variability, and you don't want to be scared if you discover you can't smell well, but that association does show up in epidemiologic studies. Many things affect the sense of smell. Blockage of the airway is one, and in children, particularly if they have obstruction from adenoids, that fortunately can be reversed with taking the adenoids out. Here you see on the left children that have on the left children that have less than 50% blockage, and you can see before the adenoidectomy and after the adenoidectomy that you improve the test score on a 12-item test. That's on that rotating test that I showed you earlier. Whereas if there's a greater than 50%, there's much more improvement that occurs, a blockage, much more improvement after the operation, as you can see there. This is work that Shima has done. I was very fortunate to be a collaborator with her on this work. She did a lot of work and tested people in the clinic along with her colleagues. Really remarkable study. And you can see the difference with these people, the acute phase of COVID-19, but basically 59 out of 60 evidenced some smell dysfunction on the Persian upset, which she developed. And about a quarter were anosmic, a third were severely microsmic, 27 had moderate loss, 13% mild loss, and only 2% were normal. And only 35% of these patients were aware of their loss before testing. And this is seen in many instances, but this clearly shows that phenomenon with COVID. Lo and behold, in another very nice study Shima and her colleagues have done, I guess it's not 2002, I think that's supposed to be 2021. But in any event, you can see we start out with COVID, and only 6% of the people in this sample were normal. But as they got out to 7 or 8 weeks, about 2/3 had gotten back to the normal range. But there's still a third that continue to have some dysfunction. And we now know from other studies that probably as many as a third of the people continue to have some degree of measurable smell loss out to more than a year. This is just being understood. And some of these studies, excuse me, are not measuring things very accurately, but nonetheless, the aggregate of the data suggests that a lot of people are going to continue to have some degree of smell loss, perhaps throughout their life. We don't know yet. By the way, this is Shima's artwork, and is really, she's a genius of color and beauty, and really made these slides. People have copied this, by the way, so I guess that's a good thing. It means that people like that. What's sort of interesting, if this was a study we did of a large number of patients that came to the center from 1980 to 2018, and we looked at the proportion that had smell loss from non-influenza viruses, post-viral olfactory dysfunction, we call that. You'll see most people come and they present mostly in April, May, in the summer months. But with influenza, they're presenting primarily in the winter months when the smell loss is present. If you look at the lower part of this, it tells you the kinds of viruses that are typically present at this time of year. The PIV3 virus, the parainfluenza virus, seems to overlap pretty nicely with the what we see with the frequency of people showing up in our clinic. But if you notice here, these were deaths that were plotted in the little blue line of COVID cases, and they tend to follow more or less what we see with the coronaviruses down below as other coronaviruses. So nobody has done this study with smell loss as a function of the time of year, but it's quite possible that this virus, the SARS virus, is following pretty much what we see with other coronaviruses in terms of the frequency of loss throughout the year. Air pollution is a risk factor for a number of neurological diseases, particularly Alzheimer's disease. And smell loss is common in highly polluted areas. Mm-hmm. This is work done in Mexico City, where even young people start to show damage into the olfactory bulb. Nanoparticles get up into the brain in highly polluted areas, and we start to see pathology related to Alzheimer's and Parkinson's disease in the olfactory bulbs of really young people. Some other interesting things, there's a gene, the APOE4 allele, it's a risk factor for, excuse me, for Alzheimer's.
Mhm.
And those individuals who had that gene were 6 to 13 times more likely than those with a different allele to exhibit olfactory bulb pathology that you see in Alzheimer's disease. So there was a serendipitous finding that those carriers were 5 times more likely than non-carriers to die by suicide as well. I'm not sure what that means, but that did appear in that particular study. Multiple sclerosis, it used to be in the old days, they thought that it wouldn't affect smell because they believed that the smell system was largely demyelinated or it didn't have myelin, but we now know the axons of mitral cells are myelinated and so on. And this was an early study we did where we showed that if you counted the plaques, so the areas of inflammatory damage that occur in multiple sclerosis, plot that against upset scores, there's a pretty good relationship that the more plaques you have, the worse your score. Whereas you go outside the brain regions that are really the major frontal brain regions for projections of the olfactory system, the temporal lobes, frontal lobes, then you don't see that relationship. Epilepsy, as many of you are aware, some people that have extreme epilepsy will have surgical interventions, but just having epilepsy, if you look at the side of the where the seizures occur, you'll find that in general, relative to controls on that side, they'll have a deficit, a smell deficit. If you look at that's with identification, the same thing shows up with thresholds. And then also we see it with other measures like odor memory and discrimination, although these tests aren't quite as reliable. So in those cases where they take out sections of the brain to control the intractable seizure activity, so-called temporal lobe resection or temporal lobectomy, they always go in and try to minimize the amount of tissue removed, particularly from the side where language is involved. So these operations are done under local anesthesia. And but you see again, the identification, this is before the surgery and after the surgery, that it typically does impact the smell function on the side where the surgery is. And you'll see that with thresholds, not quite as clearly, and also with other measures. So the bottom line is that the people start out having smell loss and they have even more smell loss when those regions of the brain are resected. Alzheimer's disease, we did among the very first studies that showed that if you have Alzheimer's, and these are people, by the way, who can do well on a picture test, so they're not so out of it, they're not so demented, they don't understand the instructions or understand what the pictures or what the smells represent. But you can see that both odor identification and odor detection is affected. Higher thresholds and poor identification scores. And some people have been arguing that somehow that thresholds are measuring only the peripheral part of the olfactory system and identification central. But as you can see here in Alzheimer's disease, as well as you saw with epilepsy, that thresholds are affected as well. So that's an old wives' tale that continues to be reported. Of course, you can get more sophisticated and look at the activation areas of the brain that light up or take up glucose metabolism, for example. You can look at labeled glucose ligands and see that in the healthy elderly, you'll see some delay, but with relative to Alzheimer's, you'll see much more decrease of activity in the brain and associated with smell function. Many studies, and I've sort of stopped it a number of years ago, 2010, but even up into that time, there are a large number of studies that show that olfactory testing can predict to some degree future cognitive decline or Alzheimer's disease. And this was pretty widespread. So generally, people who can't smell will tend to be more likely to progress to have dementia in later years. Sorry, there's so many there. It just makes the point that it's a true general phenomenon. Parkinson's disease, There are 10 early warning signs of disease, according to the National Parkinson's Foundation, and one of those is loss of smell. Actually, it's a very early sign. It occurs before the masked face and facial expression. All of these motor things come in much later. Smell, in some cases, 20 years out, will be shown. If you talk to many people with Parkinson's and ask them, can you smell? And they'll say, oh, I haven't been able to smell for years. So it is an early sign of that disease. And James Parkinson, back in 1817, when he talked about, in a brilliant essay, and when he defined this disease, he did say the senses and intellect were not injured. So he was wrong on that point if you count the sense of smell as a sense, which some people don't, unfortunately. So generally, over 90% of people with Parkinson's have demonstrable smell loss. It's more prevalent than tremor and motor signs. It's unrelated for the most part. There's some controversy about this, but to disease stage or the progression of the motor dysfunction. It's indistinguishable in terms of frequency or magnitude from that of Alzheimer's disease. Interesting, and those two diseases tend to go together. That is, if you have Parkinson's later, you're more likely to get Alzheimer's, for example. And then it's not responsive to L-DOPA or any anti-Parkinson's medications. And it's some brilliant work done a number of years ago by Brock and his associates in Germany, basically showed the first pathology of Parkinson's disease begins in the brainstem and the olfactory bulbs, and then gradually progresses to the rest of the brain, including the motor areas. So this sort of shows you that it gives a reason why probably there's smell loss early in the disease. Some olfactory tests are more sensitive to Parkinson's than even some of the modern imaging procedures, something called SPECT imaging of the dopamine transporter. This is just a radiologic way of looking at the health, if you will, of the brain in the dopaminergic areas primarily. But it turns out that these smell tests are more accurate in distinguishing between a Parkinson's patient than and a control than are some of the other widely used and very expensive measures. Also, if you look at relatives of Parkinson's patients, if they have a smell problem, they're much more likely eventually to get Parkinson's disease. They may have probably already have it at the earliest stages, but about 1/4 of those people are likely to end up with Parkinson's if they can't smell and they have a close relative with Parkinson's. What's interesting, as I pointed out, the smell loss is equivalent, though, in Alzheimer's and a number of other neurodegenerative diseases. And so, for example, if you look at Alzheimer's and Parkinson's in terms of the upset score, they're very similar. And these are Alzheimer's patients that aren't so out of it. They're very early stage, if you will, but they show basically the same degree of dysfunction as what we see with Parkinson's. And the same thing with thresholds. It's very, very similar, not much different. So if you take a whole lot of neurological disorders and you ask the question, which ones have a lot of smell loss, which ones don't, they can be ordered in terms of the scores on the upset, as you see here. And in fact, certain brain regions, and I'll talk about this in terms of the cholinergic hypothesis, but the nucleus basalis of the brain, the relative damage into that area seems to correlate with these upset scores, although the amount of pathological brain data that we have is pretty limited. But the 7 or 8 studies that we can count those cells, or the cells have been counted, correlates pretty well with what we see with the smell test scores. So what are the determinants of the smell loss? And this is the $64,000 question. People don't know for sure. I pointed out the fact that the pathology shows up early on in the olfactory system, but in many cases, I believe personally that the pathology is related to processes that occur even before that pathology. So a theory that I'm promoting or interested in and thinking about is that the neurotransmitter acetylcholine seems to be affected. The brain regions associated with acetylcholine are affected early in the diseases. And so there's a lot of sort of circumstantial evidence for this hypothesis. I mentioned here to reiterate that those disorders that have a lot of smell loss tend to have damage to the cholinergic nucleus basalis. Those that don't have much damage there. But obviously, there are other brain areas, and much more research has to be done on that. But this was an interesting study. It appeared in Brain a number of years ago. And this shows upset scores versus the amount of binding of cholinesterase in what's called a positron emission tomography study. And the more binding there of this, the better the performance. And you can see that positive relationship. However, you look at all those zeros. If you remember that I said that, by chance alone, you should get 1 out of 4 correct on the upset or 10 out of 40. And I couldn't understand, are all these Parkinson's people avoiding the correct answers? So I got in touch with the author of this paper, and he said, oh, well, we knew they couldn't smell, so we assigned zeros. So if you take those points out, it turns out there's no relationship with what's called VMAT2, which is related to dopamine, and it increases the size of the correlation related to acetylcholine. So again, the cholinergic function is a bigger player than the dopaminergic function in this case. Other evidence for an effect of acetylcholine, nicotine is a cholinergic agonist. And you can see in patients with Parkinson's disease, if you're a current smoker, you actually have a better sense of smell than if you're a past smoker or a never smoker. Of course, under controls of your current smoker, you tend to have a decrement in your smell function. But there is evidence that smoking prevents or minimizes getting Parkinson's disease, which is very interesting. Yes. There are actually it's also good for osteoporosis, apparently. So although smoking's not, you're going to die early if you're not careful, but there is evidence that nicotine can have some positive benefits. We assume it's nicotine. Also, a disorder, myasthenia gravis, which is often to be thought of just a peripheral cholinergic muscle endpoint disorder. These people are very tired all the time. We know pretty much the nature of the peripheral damage to the cholinergic system. They have the same olfactory test scores as people with Alzheimer's disease. So again, and now we know there's also central cholinergic processes going on, but that's a bit of a mystery. We don't know why for sure this is, but that's one of the things that is quite unique. And also, it's an autoimmune disorder. We now know that a lot of autoimmune disorders seem to be associated with smell loss. So that may just be the explanation of this, but we don't know. There are other measures of cholinergic function. I'm not going to get into the details. This is short latency inhibition, where with brain stimulation, magnetic stimulation, you induce a reflex, and you can inhibit that reflex by stimulating the nerve in this area. And it turns out that disorders that have cholinergic dysfunction, or at least ones we think do, that reflex is inhibited to some degree compared to those that don't. So this is, again, sort of evidence that there's a cholinergic role in this whole process. Animal studies, nicotine prevents, to a large degree, the smell loss that's induced by a neurotoxin, a pro-neurotoxin called MPTP. Mice that are injected with that will have damage to the dopaminergic system. But if they've been given nicotine, that protects against that damage. So again, it suggests that the cholinergic system somehow is protecting against that damage. So I'm at the end of my presentation. I want to thank you for your attention, and I'll smell you around. Thank you.
Thank you so much. Thank you.
Sure.
It's incredible what you have done, Dr. Doty, and your research was so eye-opening and so beautiful. But before I go to Dr. Alavi, I have a question. I have several questions, but I just asked one. are there any ways to prevent, the loss of smell and taste?
Stay away from children because it's viruses that do a lot of the work on it, and it's cumulative. If you look at the olfactory epithelium of a rat that's in a filtered cage, then it doesn't show age-related changes. Whereas if you look at a rat that's in a non-filtered cage, it's exposed to the virus, the environment, and so on, that cumulative effect occurs. And we know from EM studies that there's constant, every time we get a bad cold, every time we get exposed to pollution, there's little islands of damage we call metaplasia that enter in. And by the time we're in our 50s and 60s, our epithelium is not pristine. It's all pockmarked, much like cheesecloth. So we know the environment is a major player. So obviously, if we were all wearing masks from the age of 3, we probably wouldn't see a lot of the pathology. There are other reasons for age-related pathology. For example, the holes through which those nerve fibers project between the nasal cavity and the brain cavity tend to ossify over. And then also we see, central pathology, and there's other reasons as well, changes in vasculature and things of this sort. But viruses are the big player, and if you can avoid viruses, so when you're around your children or grandchildren or great-grandchildren, wear a mask. And it may scare the child, but it will, may protect you from continuing to get colds and upper respiratory infections. In terms of other preventive measures, we do know that certain vitamin deficiencies can damage the smell system. But if you're getting normal diet and you take a multivitamin or whatever, you can protect against that. It's relatively rare, but it does occur if you have very major zinc deficiency, for example, or some other deficiencies. I think keeping an active mind, exercise. There's studies that show that people who exercise regularly are less likely not only to get Alzheimer's disease, but less likely to have smell loss. So those are the major ways that we know at the present time. Smoking cigarettes, I wouldn't recommend, but maybe in some cases that might be helpful as well, but I don't recommend that.
Incredible. Thank you so much. But before we go to Dr. Alavi, let us hear a few words from Dr. Moin, who has also, done research on smell and taste. Dr. Moin, Dr. Garrison, can you unmute Shima? Do you see Shima? Shima, raise your hand.
Yeah, if people just yeah.
Good.
She's unmuted.
Okay, Dr. Moin, can you please tell us a few words about what you have done?
Yeah, sure. Thank you, Dr. Doty. It was a great pleasure for me to be part of his research team. And he had been very encouraging to me and supportive all the time that I was in Iran, and also supported me coming here.
Noise is coming from your house. Yes. Is the television on, Shima? Something?
I'm in the office. No, it's not.
Oh, you're in the office. Yeah. Mm-hmm. Yeah. Some other people are talking. Yeah. Okay.
Well, the thing is that I would like to share one slide if it.
Mm-hmm.
So when I went back to Iran and started decided to do the research on smell, I figured out that we don't really have a test for smell in Iran. So that's when I started working with Dr. Doty to revise the smell test for Iranian population, because we know that smelling different odors with changes with experience, and your previous history of exposure to different odorants can affect your identification rate for those odorants. And I was somehow very lucky that once this test was ready and we had collected the data from the normal population, then SARS-CoV-2 started to spread in Iran. And Iran was the third country after China. At the same time with Italy, we were hit with the virus. Then I had another chance. Then I had the background in medicine, so I had many colleagues in the hospital. And some of them called me since they knew that I had started working on the olfaction and told me that some of our patients who have the disease claim that they can't feel the smell. So that's when I called Dr. Doty again and said, I think I've got a conflict of interest with the disease. It seems that there is something in SARS-CoV-2 infection and we have to test these people. So that's when we quickly started testing people who were identified with the SARS-CoV-2 infection and were admitted in the hospital. So they were in the severe phase of the disease. And we could show that there is small dysfunction in almost all of them. And our study was the first one that used the objective testing for such patients. And we could compare it with the normal population. Then we could follow up, as Dr. Doty presented in his talk, with these patients over 2 months to see what percentage of them recover from the small dysfunction. And the good news was that over 2/3 of them recover without any intervention. But the bad news was that still 1/3 of them have some degree of dysfunction, and we still don't have any kind of treatment or therapy for these people. And that's a kind of problem because most of the grant agencies and the institutions that cover for research, they haven't put that much of money in order to find a kind of therapy for small dysfunction over the years because they thought it's not important and a very few population may have it. But now with the COVID, we know that it's going to be a problem for people who have recovered from the disease, probably over 1 year. Less than 1/3, but still a huge number of them will be affected by small dysfunction. So that's the main thing that many of the patient advocacy groups are now trying to push. For instance, NIH or other governmental agencies to invest in finding some treatment for that. Okay, I don't take much more of your time so that you could ask the questions from Dr. Doty. And thank you for giving me nice opportunity. Again, thank